Hi students, welcome to Neet online biology classes. So this is the fifth day of human reproduction chapter. In last four video we covered the pre-fertilization structures. In that one, the male reproductive structure we covered. Yes. Then after the female reproductive structure we covered. Now the time comes to cover the pre-fertilization events. Pre-fertilization events means the main is a gametogenesis. Yes. So gametogenesis means it is a formation of the haploid gamete in respective gonads is called the gametogenesis. Now, gamete means it is a cell which contains single set of chromosomes. Gonads means these are the special organs where the gametogenesis occurs. Okay? So, gametogenesis means so it is a formation of formation of haploid gametes. Gametes means sperms in male and ovum in female. The haploid gametes in respective gonads means testis in male and ovary in female is called as gametogenesis. Now, it is a formation of haploid gamete. So, gametes are always haploid. So, either it is a spermatozoa or the sperms in male one, ovum in female, in respective gonads. Gonads means the testis in the male and the ovary in the female. So, it is called the gametogenesis. Yes. Now, the major question, which type of cell division will occur in this one? So, this is a haploid one, but when you take the gonads, the specifically testis in the male and the ovary in the female, in that special cells are there. These are called the primary germ cells or the primordial germ cells are called, which are the diploid. Yes. So, when you take this, any gonads, so either testis or the ovary, so gonads have primordial germ cells which are diploid in nature. Yes, so these are the diploid in nature. So these primordial germ cells produces. So these gonads are the primordial germ cells which produce gonial cells which are also diploid. Here, so primordial germ cells are diploid, gonial cells also diploid. So means both are diploid. In between this one mitosis occur here. So gonial cell. So these gonial cell means cells means in male it is called as a spermatogonial cell. Yes, in male, spermatogonial cell in male and oogonial cells in female. Yes, primordial germ cells are diploid, they produce gonial cell. So these gonial cells specifically called the spermatogonial cell in the male one and oogonial cell in the female. Yeah. So these gonial cells undergo meiosis and produce gametes. Gametes means, so they are nothing but the sperms in the male and the ovum in the female. Yes. So means if you want to see the schematic diagram, so the gonads are there. Yes. In this gonad, primordial germ cells are there. So these are the diploid in nature. The primordial germ cell undergo the division and they produce the gonial cell. Here, so these gonial cells are in male. It is a spermatogonia. In female, it is a oogonia cell. Yes, so these are diploid. Gonial cells also diploid. In that one, in the male, it is called a spermatogonia cell, and in female, it is called a oogonia cell. Now, these cells undergo meiosis and produce spermatozoa. Yes, in this one, the oogonia undergo produce only one ovum. Yes, so you can see the difference. In the male one, the spermatogonia produces the 4 spermatozoa. In oogonia means female one, oogonia means oogonial cell produce only one ovum. So small polar bodies are produced. So the difference between these two we are going to cover in detail. Okay? So ultimately this is only the definition of gametogenesis. Means the formation of the sperm in the male and formation of the ovum in the female in respective gonad is called as a gametogenesis. Clear? Now, in male, this one, the formation of the spermatozoa from spermatogonia, this is particularly called as a spermatogenesis in male. In female, it is called as a oogenesis. Means in male, the gametogenesis is particularly called as spermatogenesis. In female, it is called the oogenesis. Now, we first cover the spermatogenesis in detail. Later on, we cover the oogenesis. Clear? So, first topic is the spermatogenesis. So, first one is the spermatogenesis. Spermatogenesis. So, the definition is simple one. It is nothing but the formation of haploid, motile and haploid motile spermatozoa from spermatogonial cell is called as spermatogenesis. Yeah. So we have to produce spermatozoa from spermatogonia. So this is called the spermatogenesis. Yeah. So here both mitosis and meiosis type of division take place. Ultimately, and take this one, it is further divided 3 major: spermatogenesis, spermiogenesis, and the spermiation is the later part. Yeah. So spermatogenesis means this takes place inside the seminiferous state. When you take the TS of testis one, So this is one testicular lobule. Inside 1 to 4 seminiferous tubules are present here. So the spermatogenesis takes place inside the seminiferous tubule here. So in between the seminiferous tubule, the interstitial space is there, and some special cells are present in this one. These are nothing but the interstitial cell or the lytic cell. Yes. So this is the testicular lobule. So this is a testicular lobule. This in the seminiferous tubule. Seminiferous tubule. The space is called as a interstitial space. Interstitial space. Yes, the cells which are present in between the space is called the, these are the lytic cells. Lytic cell. Yes. So now seminiferous tubule, inside the seminiferous tubule, as we know, they have the two types of cell. One is a primary germ cell. Yes. And one more cell here, that is called the Sertoli cell or the nurse cell. These are the Sertoli cells. Detail we will go. Yes. Now, so this is a testicular lobule. Inside 1 to 4 seminiferous tubules are there. So in between that one interstitial space is there. Here some special cells are there. These are called the lytic cell or the interstitial cell. Inside the seminiferous tubule, two types of main cells are there. One is the primary germ cell and the Sertoli cell. Yes. Now the spermatogenesis takes place inside this one. Now how the event means? Now pictorial will show. So this is the primordial germ cell. Yes, primordial germ cell, primordial germ cell. This undergo division and produce the spermatogonia. So these are spermatogonia. Spermatogonia. Yes. So these are diploid in nature. They contain two set of chromosomes. Means one chromosome, this is another chromosome. Yes, these are the spermatogonia. Now these spermatogonia undergo repeated mitotic division and produce large number of spermatogonia cells. They undergo repeated mitotic division and produce large number of spermatogonia. So this is mitotic division and they contain two set of chromosomes. Here the chromosome number will not differ. So every spermatogonia contains two set of chromosomes. This is also diploid and these cells also diploid. Every spermatogonia have two set of chromosomes. So these are still spermatogonia only. Yes. So now which type of division occurs? So this is a mitotic division. Here mitotic division means polygon will remain same. Now this, out of this one, some of the spermatogonia will act as a reservoir, some will undergo the next process. So out of this one spermatogonia will enlarge and produce primary spermatocyte. So this is a little bit enlarged. So almost size of the cell doubles Now this cell is called a primary spermatocyte. Only few of them. So this is primary spermatocyte. This is primary spermatocyte. Yes. Now this phase is a growth phase. Here, growth phase. So due to the accumulation of the means, due to the increase in size, that's why it is called the growth phase. Yes. So this is the primary spermatocyte. This is also diploid. The spermatogonia is diploid. Primary spermatocyte is also diploid. Now this primary spermatocyte undergo meiosis. So it undergo first meiosis and produce two haploid cells. So this undergo first meiotic division where actual reduction of chromosome number occur. Now this contain only single set of chromosome. Yes. So primary spermatocyte undergo first meiotic division, produce secondary spermatocyte. Secondary spermatocyte. Yes. So these are haploid in nature, and this division is meiosis 1. Meiosis 1. Immediately after, without following the replication, this secondary spermatocyte undergoes second set of meiotic division and produce 4 cells. So this is the second meiotic division and produced 4 haploid cells. So this is the actual reduction division where the 2n turn into n1. So second spermatocyte haploid. This is the second meiotic division, meiosis 2. Yes, immediately. Now these cells are called as spermatids, which contain single set of chromosome. Yes, every cell has again contain single set of chromosome. So these are called as a spermatids. Spermatids. Yes. Now these two divisions, meiosis 1 and meiosis 2, are collectively called as a phase of maturation. Now complete process can be divided into 3 phases. First one is the phase of multiplication, where only mitotic division occurs and produce large number of spermatogonia. Second one is the phase of growth, where only some of the primary spermatocyte enlarges and turn into primary spermatocyte. Third one is the phase of maturation, where the meiotic division occurs, meiosis 1 and meiosis 2, and produce the spermatids. So for convenience, it is divided into 3 phases. Now phases I write here. So this is the first one is the phase of multiplication. Yes, produce large number of spermatogonia. Second one, or the proliferation also called phase of proliferation. Yes. Second one is the phase of growth. Yes. Third one, where the actual meiotic division occurs, meiosis 1 and meiosis 2. This phase is called as a phase of maturation. Means spermatogenesis is divided into phase of multiplication, phase of growth, and phase of maturation. After the completion of phase of maturation, 4 spermatids are produced. But these are not spermatozoa. Yes, so this is the spermatogenesis. So next one is the spermiogenesis. Spermiogenesis means these spermatozoa undergo differentiation and produce functional sperms. So each spermatid has potential to develop into spermatozoa. So these spermatids are transformed into special structure. These are called the spermatozoa. Now, spermiogenesis means the conversion of non-specialized, non-motile, spherical spermatids into highly specialized motile spermatozoa is called as spermiogenesis. These structures are nothing but the spermatozoa. Yes, and this transformation from the spermatid to spermatozoa is called as a spermiogenesis. Yes, this transformation takes place on the surface of the Sertoli cell. So this is the only spermatogenesis. Now transformation is the next one, that is the spermiogenesis. So pictorially we have to show this one. So this is the spermatogenesis. Now this process actually began only after attaining the puberty. Means this complete process starts at the age of 12 to 14 age, when the human male is sexually mature. Means when the particular hormones are secreted, and then only this process starts. Have understood? Now here ploidy is important. Spermatids also haploid. Yes, spermatozoa is also haploid. Here only up to the primary spermatocytes are the diploid. Spermatogonia is diploid. This undergo multiplication phase, produce large number of spermatogonia. This is also diploid. So it undergo growth phase. So still chromosome number remain the same. Primary spermatocyte is also diploid. Primary spermatocyte undergo first meiotic region, produce the secondary spermatocyte. Now secondary spermatocytes are the haploid one. This undergo second meiotic region and produce the 4 spermatids. These are haploid, and spermatozoa is also haploid. This process is a continuous process. Continuous means it will not halt anywhere. Yeah, this will continue. This will produce continuously. Now every primary spermatocyte produce 4 spermatozoa. Yes. So if you want to draw the figure, how it occurs in the seminiferous means. So here I will show. So this is the definition of gametogenesis. So now we are going to do only spermatogenesis. So so this is the testicular lobule. So these are the seminiferous tubule. Yes. So in between this one, interstitial space is there, where the Leydig cell is present. Yes. So inside the seminiferous tubule, 2 types of cells are there: primordial cell or the primary germ cell. Yes. These undergo division, and in between that one, some special cells are there. These are the Sertoli cell. These red colors are the Sertoli cell or the nurse cell or the sustentacular cell. So this is the testicular lobule. So these are the seminiferous tubule. Inside that one, 2 types of cells are there: primordial germ cell and Sertoli cell. Yes, the germinal epithelium consists of 2 types of cells: primordial germ cell and Sertoli cell. The cells which are present in between this seminiferous tubule, these blue color cells, these are called as the Leydig cell. Yes, Sertoli cell is also called as the nurse cell, sustentacular cell. Leydig cell is called as the interstitial cell. Now, what happens if you enlarge this figure? So this is the germinal epithelium. Yes, outside here Leydig cells are present. Yes. So these are the primordial germ cell. Yes. So this primordial germ cell undergo repeated division produce the spermatogonia. So this produce spermatogonia undergo mitotic division. These are the spermatogonia. Yes. Out of this one, some of the spermatogonia enlarges and turn into primary spermatocyte. These are the primary spermatocyte enlarged. Now these primary spermatocyte undergo first meiotic division produce the secondary spermatocyte. These are the secondary spermatocyte. These secondary spermatocyte undergo second meiotic division produce the spermatids. 4 spermatids this side and 4 spermatids this side. These are the spermatids. Yes. Now these spermatids gradually transform into spermatozoa. These are gradual transformed spermatozoa. Yes. In between that one, so this large cell is this is a Sertoli cell. This is a red color cell which I am showing here. So this is a nurse cell or sustentacular cell or the Sertoli cell. This is a Sertoli cell. Now, this complete process is the spermatogenesis. Now these immature spermatozoa attack and turn into mature spermatozoa. These are the spermatozoa. Yes. So this is a spermatogonia. Spermatogonia. This is a primary spermatocyte. This is secondary spermatocyte. So these are spermatids. These tadpole-like structure are called as spermatozoa. Yes. Now, how the hormonal regulation occurs? How this process starts? Here. So what happened? This is the this is the Sertoli cell. So this is the Sertoli cell. Yes. So the function of Sertoli cell is mainly due to the spermiogenesis, means the conversion of the spherical non-motile spermatid into the tadpole-like spatial motile spermatozoa occur on the surface of the Sertoli cell only. Here the Sertoli cell plays a very important role. Means when you take a spermiogenesis, here many changes occur. What are the changes means? So these spermatids are spherical structure, spermatozoa are the tadpole structure. So during the spermiogenesis, what occurs means condensation of nucleus occur. The nucleus became very compactly arranged one. So that the sperm and where the acrosome develop. Here acrosome formation take place from the Golgi bodies one. The centriole divides and turn into the proximal centriole and the distal centriole, and the accumulation of the microtubules occur just behind the nucleus one, and ultimately the cytoplasma contain the reduced one. So this spherical structure turn into the tadpole-like structure that is called as spermatozoa. The major changes occur in the spermiogenesis only. This complete process takes place on the lateral side of this Sertoli cell. This Sertoli cell will enclose the spermatogonia, spermatocyte and spermatids and also the spermatozoa also. And this Sertoli cell will provide a nourishment for this process. That's why these are called as a nurse cell. Yes, these Sertoli cell will function under the influence of the FSH hormone. Yes, follicle stimulating hormone, which is secreted from the pituitary gland, and this Sertoli cell produce the androgen binding protein. Here, so due to that one, whatever the testosterone produced from this Leydig cell will accumulate, means androgen will accumulate here. So due to that one, spermatogenesis, spermiogenesis occur here. That hormonal regulation I will draw. So one more activity, this Sertoli cell also produce inhibin. Inhibin means if the follicle stimulating hormone is repeatedly produced, to stop that feedback, this produces the inhibin that will produce a negative feedback. From the pituitary gland, the production of FSH stops. So the complete the hormonal regulation, everything occurs inside this testicular lobule. So this is the sectional view of the seminiferous tubule where the spermatogenesis, spermiogenesis occur. Yes. So after spermiogenesis, when the spermatozoa are completely mature, yes, these spermatozoa are released into the lumen of this seminiferous tubule. Means here the spermatozoa are released one. Yes, that process is called as a spermiation. Means here three terms are there. One is a spermatogenesis, second one is a spermiogenesis, third one is a spermiation. Yes, just try to understand what are the actual process. Spermatogenesis means it is a process where the production of the specialized spermatozoa from non-specialized primordial germ cell or the spermatogonia. This complete process is called the spermatogenesis. Spermatogenesis again divided to phase of multiplication, phase of growth, phase of maturation. Yes, but in that only, spermiogenesis means the transformation of spherical, non-motile and the non-specialized spermatids into tadpole-like, motile, specialized spermatozoa is called spermiogenesis. That spermiogenesis takes place on the surface of this Sertoli cell. Yes? Spermiogenesis finished one, then the complete mature spermatozoa have to release from this Sertoli cell. The release of spermatozoa from the surface of Sertoli cell into the lumen of this seminiferous tubule, that is called the spermiation. Yes? So all these definitions are given in the NCERT textbook. Spermatogenesis, spermiogenesis and spermiation. So now we are going to cover how the hormonal regulation occurs to control this complete process. Clear? So here two types of cells are there. One is a Sertoli cell, you already know. So this is the Leydig cell. Yes? So just I will draw hormonal regulation on this side of it, how the hormonal regulation occurs and how the Sertoli cell plays a very important role in the spermiogenesis process. So first one, hormonal regulation. Hormonal regulation means first comes hypothalamus. Yes, this hypothalamus will produce main hormone, that is gonadotropin releasing hormone, that is GnRH, gonadotropin releasing hormone secreted from the hypothalamus. This will act on pituitary gland. This gonadotropin releasing hormone act on the pituitary gland. So from this pituitary gland, specifically from anterior lobe, anterior pituitary, this will produce two hormones. One is luteinizing hormone, that is LH. Second one is follicle stimulating hormone, that is nothing but FSH. Yes, this LH is act on, this LH will act on Leydig cell. Yes, this Leydig cell will produce, androgen or testosterone. Yes, this Leydig cell will produce the androgen. This androgen means this Leydig cell will produce the androgen here. Now this Sertoli cell will produce androgen binding protein. This is androgen binding protein. Yeah. So due to presence of this androgen binding protein, the concentration of androgen will increase near the Sertoli cell. Yes, the androgen is produced from the Leydig cell. This androgen will accumulate near the Sertoli cell. Okay? Under the influence of this androgen and follicle stimulating hormone, this follicle stimulating hormone will act on Sertoli cell. Yeah. So the Sertoli cell under the influence of FSH, so complete spermatogenesis process will take place. Yes, and spermiogenesis also occur on this Sertoli cell. So this Sertoli cell will also produce androgen binding protein and one more element, that is a inhibin. Clear? If the inhibin will produce negative feedback, means it will stop the production of FSH. Now try to understand. GnRH produced from from hypothalamus. This GnRH act on pituitary gland. Pituitary gland produce LH and FSH. LH will act on Leydig cell. Yes, Leydig cell will produce the androgen. This will produce the secondary sexual character of the male also. Now FSH will act on Sertoli cell. Yes, the Sertoli cell will produce the ABP protein, androgen binding protein. Due to that one, the androgen concentration increases here. Also it produces the inhibin one. So if the FSH production is more one, this will stop the further production of inhibin. Means this will reduce the FSH production. Due to that one, the process will stop there. So the Sertoli cell plays a very important role in the spermiogenesis. Just try again. What the changes occur in spermiogenesis means? So spermiogenesis means now role of Sertoli cell. Yes, I have to know it produces the ABP also and inhibin also. Role of Sertoli cell. I have to know spermiogenesis process. Spermiogenesis means, so this is a spermatid and this is a spermatozoa. We will take this spermatozoa. Yes, it is mature cell. So see the structural difference. When you take the spermiogenesis is nothing but the transformation of the spherical non-motile spermatid into the motile tadpole-like specialized structure that is called the spermatozoa. What are the changes take place means here condensation of nucleus occurs. Yes, nucleus is highly condensed. This is a nucleus where the chromosomes and the nucleus is very difficult to distinguish. The nucleus is highly compact. Yes, second one, formation of acrosome. the tip of the spermatozoa, here the special structure is there, that is called the acrosome. The formation of acrosome from Golgi bodies. From Golgi body. Here, so this acrosome contains the hydrolytic enzyme which is required for the fertilization. So this acrosome produced in the spermatozoa. So apart from this, division of centriole, occur. So centriole is present at the middle region, the proximal centriole and the distal centriole. So next, accumulation of microtubules behind the nucleus. Yes, so these microtubules are required for the division of the nucleus. So in the oocyte. So this is required for the further process. That accumulation of microtubules occur just behind the nucleus one. Then main means loss of cytoplasm. When you compare this structure and this structure, in spermatids, lot of cytoplasm is there, but in the spermatozoa, cytoplasm is very small amount. Here, the loss of the cytoplasm takes place. Whatever the cytoplasma loss occurs, that is absorbed by this means by the Sertoli cell. Yes, that is spermiogenesis occur on the surface of the Sertoli cell. That's why to form the mature spermatozoa from the non-specialized spermatogonium, this complete process almost takes 64 days. Yes, so this takes almost on an average 64 days to produce the spermatozoa from this spermatogonium. Yeah. Now, what is the role of Sertoli cell means? This produces the androgen binding protein, where the androgen concentration increases. This produces inhibin also, and whatever the cytoplasma is lost from the spermatid during the production of spermatozoa, that is also absorbed from this Sertoli cell. And this Sertoli cell will produce the fluid. Due to that one, the spermatozoa are easily show the movement also. So this is about the hormonal regulation and the spermiogenesis. What the changes take place during the spermiogenesis. So just read the hormonal regulation in detail. Yes, hypothalamus produces the gonadotropin releasing hormone. This will act on the pituitary gland. Anterior pituitary produces the two hormones. They are the pituitary hormone LH and the FSH. LH will act on the Leydig cell, which is present in between the seminiferous tubule. This Leydig cell produces the androgen. Based on this one, the androgen is produced from the Leydig cell only under the influence of LH. The FSH will act on the Sertoli cell. The Sertoli cell plays a very important role in the spermatogenesis also, spermiogenesis also. So this Sertoli cell will act as a nurse cell. This produces the ABP, androgen binding protein, and it also produces the inhibin. And one more, this Sertoli cell also produces the Mullerian degenerating hormones also. Clear? So this is about the hormonal regulation and the spermiogenesis. Now we are going to cover the in detail structure of the spermatozoa or the human sperm. Yes? Yes, good. Now this is the last topic of today's video, that is nothing but the human sperm. Here, the figure is there. So human sperm, so it is almost 40 to 60 micrometer length. Yes, micrometer in length one. Total length almost 45, 50 or 60 micrometer length. So it is divided into head and the tail. Yes. So when you take the head piece, the tip of this one, it is covered with the acrosome. It is a special structure. Just I have put the acrosome produced from the Golgi bodies one. So this acrosome consists of the hydrolytic enzyme. For example, hyaluronidase, zonalizing, or the corona penetrating enzyme. So the acrosome plays a very important role in the penetration of sperm into the ovum here. So the acrosome, so it consists of hydrolytic enzymes. So this will cover the nucleus of this sperm one, partially, the anterior region. So this is the nucleus. Yes, where the chromosomes are highly condensed. It is highly difficult to distinguish where the nucleolus and where the chromosomes are present. So this transformation takes place during the spermiogenesis only. I have tried to the condensation of the nucleus takes place. Nucleus is present in the head region. So this is the head region. The acrosome and the nucleus collectively comes under the head region. So this is almost 4 to 5 micrometer length. Here, the head is followed by the tail region. So in between that, this region is called as the neck. So little bit constricted region. This is called as the neck. So almost it is a 0.5 micrometer length one. So in the neck region only, the centrioles are. So two centrioles are there. This red color upper. This is called as a proximal centriole, and this is like this one. Proximal centriole is arranged like this, and distal centriole is perpendicular to almost to the this centriole. So this is proximal centriole, distal centriole. From this centriole, this axon is produced. Here, so nucleus and acrosome both are covered by the plasma membrane. This is a plasma membrane. The same plasma membrane will cover the complete structure of this chromosome. Here, so head is consists of nucleus and acrosome. So both are covered by the nuclear membrane, means plasma membrane. The nucleus is covered by the, as usual, double membrane, nuclear membrane. Here. So the neck region where the centrioles are there, proximal centriole and distal centriole. So both are 90-degree angle to each other one. So from the distal centriole, axon is produced. The neck is followed by this portion. This portion is nothing but the middle piece. So this is the middle piece one. Yes. So this is functionally very important. The middle piece is almost 5 to 7 micrometer. This is 5 to 7 micrometer. Yes. So middle piece. So in the center of this one, when you take the transverse section of this middle piece also, main part. So it will appear like this one. So this red color centriole axon is central part. So this the central red color portion. This is called as a axon, which is in the form of 9 plus 2 arrangement, where the 2 microtubules in the center and the 9 microtubules, doublet microtubules are present toward the periphery. So this axon is made up of microtubules, which is made up of tubulin. Yes. So this is the central axon which passes through the middle piece also and the tail piece. This is the axon. So this is followed by the 9 black color dot. This is the accessory fibers. Accessory fiber. This will give the strength to this middle piece also. These are the accessory fiber. This accessory fiber is surrounded by this green color portion. So this is a very important one. So here mitochondria arranged in the spherical manner. So this triangular structure. So these are nothing but mitochondria. Mitochondria. It is specifically called as a Nebenker. Yes. So mitochondria are spirally arranged around this axon. This mitochondria is responsible for the anaerobic respiration and produce the ATP and provide the energy for the motility. So this mitochondrial structure is specifically called as a Nebenker. Here, Nebenker. So which is present in this one. So surrounding that one, plasma membrane is there. So this complete structure is the middle piece. Yes. At the end of the middle piece, this black color structure, these are the centriole ring or annuals. Yes, at the end of the middle piece, the centriole ring is. So this is the middle piece measuring about 5 to 7 micrometer. the middle piece is followed by the tail region. So in the tail region also, so the initial portion is called the principal piece and last piece is called the end piece. So this region is called as the principal piece and this last portion is called the end piece. Yes. So what is the difference between middle piece and the principal piece means? So here also the axoneme is arranged with the 9 plus square element only in the center, which is followed by the 9 accessory fibers. But here mitochondria is absent. The mitochondria is not there. But this axoneme is followed by the fibrous sheath. Fibrous sheath, which is present all over, and that is followed by the plasma membrane. Can you differentiate? In the middle piece, axoneme is there. Accessory fibers are there. Mitochondria is spirally arranged, that is called the ribbon curve, which is followed by the plasma membrane. Here the mitochondria is absent, instead of that the fibrous sheath is there. So this principal piece almost 30 to 35 micrometer in length one, which is covered by the plasma membrane. The end piece is the last portion where the plasma membrane is absent. Yes, when to complete structure one, here the amount of cytoplasma is very less. So which is lost during the spermiogenesis. It contains acrosome, nucleus in the portion of head. Neck is a connection between the head and the middle piece. The middle, in the neck region, proximal and distal centrum is there. The middle piece where the axoneme is in the center, which is followed by the accessory fiber, and the mitochondria is arranged in the form of Neven curve. So middle piece is followed by the tail region. So principal piece is almost 30 to 35 micrometer. The end piece is the last where the plasma membrane is absent. So this is the human sperm. Clear? So roughly the human beings produce 10 raised to 9 to 10 raised to 10 numbers of sperm. Almost 50 to 150 million sperms every day. Clear? These sperms are stored and undergo maturation either in the epididymis or the vasa deferens one. So when ejaculation occurs or the insemination occurs, these sperms are loaded into the female reproductive tract. Yes. So here, when you take this acrosome contains the hydrolytic enzyme. It will come during the fertilization of one. The acrosome contains many hydrolytic enzymes like hyaluronidase, acrozyme, or the coronapenetrating enzyme. Here some special receptor proteins also there. So that will show the compatibility with the plasma membrane of the ovum. Means the sperm plasma membrane consists of the anti-fertilizer or the binding protein. Yes. So this is about the human sperm. Next topic is comes under the gametogenesis only. That is nothing but the oogenesis. That oogenesis show the means that is completely different from the spermatogenesis. In next class we are going to cover the next topic, that is nothing but oogenesis and structure of the ovum. Yeah. Till then, just do your hard work and also smart.